here and giving me the opportunity to present some of the work that we're doing in my lab at MIT. So I'd like to start a little bit about talking about what we have expertise in. We work with polymers and drug delivery-based systems that are able to deliver and stabilize molecules such as biologics, and really for important public health applications. Today, so we work across various applications, both in the vaccine space, the cancer therapeutic space, small molecules, as well. But today I'd like to focus on two technologies that we developed and that are being commercialized for use in humans. So I'll start with the motivation for some of our work. We have great data now on from the COVID, unfortunately from the COVID-19 pandemic. But what we've see really clearly now is that the ability for people and patients to come back for additional booster doses in the area of vaccination is certainly declines. And this data is prevalent across the world. But more importantly, this has existed for quite a long time, especially in developing world countries, the low and middle income countries. And I think this slide sort of emphasizes this, that even with COVID-19 data, obviously we see areas which on the right are very light, that are under-vaccinated. So my motivation in this space resulted from really seeing how many children, I think it's on the order of globally about 5 million children die annually from under-immunization. So the vaccines are available to prevent these adverse events, but unfortunately these children aren't able to gain all of the shots required to be vaccinated. So one idea that we had to address this issue from a technical standpoint, we do know that many of these children, and as we saw in the COVID case, do get at least one shot. And so the idea is, can we create something, and ideally a platform technology, because as we know, there are many vaccinations that are necessary for childhood diseases, preventing childhood diseases. And so is there a way to create a platform where we can give one injection, and in that injection, one would potentially not only get all of the booster doses required, an example of polio, you have to receive 3 shots over a couple of months, but also you could get different vaccines that could be delivered at the same time. And the technology will allow it to stay under the skin, as illustrated here on the bottom. So the vaccine is encapsulated in a material that could stay under your skin, and it would be pre-programmed to release at a particular point in time, depending on the vaccine of interest. So that was the idea, but fundamentally in the area of drug delivery, this was really difficult to achieve, and we failed many, many, many times, and actually that was a good thing, as you all know, because that enabled us to get really good patent protection for the technology once we did figure out how to make it work. But in the drug delivery field, as you see on the left, and even whether they're nanoparticle or microparticles, for example, the mRNA, LNP COVID vaccines, the drug or the vaccine is distributed throughout the particle, both on the surface and inside the particle. And what happens is when these things are injected, the drug of interest or antigen, again, is released and is seen by the body right away after the injection. And then there's usually a secondary point where these molecules slowly diffuse through passive diffusion from these particles. And the scale of that can vary. It can be hours, it can be days, it can even be months in some instances. But the issue is that it's a passive process, and it's really difficult to prevent that initial burst. And so our hypothesis was actually very simple. If we create a structure analogous to an egg, where the core has the vaccine or the drug of interest, and then the shell is really what mitigates the release of that drug, and only then, only when this shell is ruptured, if you will, the egg yolk or the drug of interest is released. So that was the idea. Unfortunately, we failed, as I mentioned many times, to be able to create this from materials that are FDA approved and safe. As we all know, vaccination is given to healthy individuals, so that's and children and even infants, and so that's very important for the technology to be translatable. And then really, too, to be able to allow the particles to be injected. So be small enough and really control that structure at a really small scale, smaller than a grain of salt. And so we did come up with a method. We ended up borrowing a lot of technological tools from the microelectronics industry and soft lithography. And so this is a video illustrating, we ended up creating these, they ended up being cubic cups, which can be made from any thermoplastic material, which can be filled robotically with a piezoelectric nozzle. So the example you see here is something that you would find in your inkjet printer. And then we figured out a way to align and seal the particles with that same material. And then these little particles can be isolated and mixed together with other ones. And I'll go into some details about how that technology works. Here's another video. This is an actual slide containing some of these little particles. They're being filled with a dye in this case, but just to illustrate the sort of scalability and the ability to really fill these particles with essentially any drug of interest. And then finally, if you look at the bottom particle here in the center of the screen, you'll see there's a diffraction pattern. So this is an optical image showing that we can optically follow. And so you could use AI and other factory-based imaging analytics to ensure that the particle is sealed, because for certain applications, we wouldn't want to see a premature release of the drug. So with this technology, I just want to kind of summarize. We wanted to use FDA-approved materials, so we focused a lot. Again, any thermoplastic material could be integrated into this technology, but we focused on using PLGAs, which are the type of materials that you use in resorbable sutures. So if you had any dental work done or minor surgery, and you don't have to have the stitches removed because they degrade on their own, these are the materials that we used, really a long history of a safety record in the clinic. And again, these are just some more images to show you what the particles look like under electron microscope there on the left, an optical image in the middle. And to really understand that you can make these in these large arrays, I actually have a glass slide here, so one could put about 1,500 particles on this slide. And depending on the dosing, you need maybe in the tens or hundreds, depending on the application. And really, this is the kind of the most important aspect of the technology. What you see here on the left, all of those vertical lines are different release points of these particles in vivo. And you can see that we have very beautiful data where we can control when the particle releases, and it releases in a very fast burst, if you will. You can see we can resolve it on the order of days, weeks, months, and that last point in the graph is 6 months out. So we really have this beautiful control. And all of the materials that you see listed there, they're all these lactic glycolic acid polymers. What we vary is the chemistry in the backbone of these materials and the end chemistry of the polymer chains. We can also dope the particles with various buffers to also ensure or control when this degradation occurs and the drug or the vaccine is released. And I want to emphasize that this allows you to then mix all of these particles together, so you could potentially create a vaccine that gives you this kind of kinetics. And then we've shown in various models, and I'll just go, for the sake of time, go through these very quickly, but we showed that you could use this with various vaccine models and also various types of molecules. So for example, we've shown that with a hepatitis B vaccine, which is a recombinant protein, we were able to formulate both the vaccine and a CpG adjuvant. And as you can see in that graph there in the bottom left, the red line is our formulation, so single shot of this technology. And then our black line is a comparable control, so 2 bolus separate injections. And you can see that the immune responses in this case are equivalent. In addition to that, you can see that in this case, a solid, so these are solid formulations, unlike the liquid things that you usually see with many vaccines. And so in solid forms, because the molecules are immobilized in the matrix, we do see quite nice stability at room temperature in a lot of our formulations that we work with. And that's illustrated there. Another example is the inactivated polio vaccine. And in this case, it's actually, inactivated polio vaccine requires 3 different serotypes, and it's a 1 and a 2 month release point. So you need quite a bit of vaccine to deliver, if you will. And not only that, it is very sensitive, this particular vaccine, to a drop in pH. And as you can imagine, these particular materials degrade into lactic and glycolic acid, which will lower the local pH. And so we spent quite a bit of time, which is illustrated here in the upper left, engineering a 2nd-generation particle that has inherent in its platform a stabilization tool to prevent low pH buildup in the particle. And I'm happy to talk about that in more detail later. But what I wanted to emphasize here, and especially emphasize our failure, if you look at these graphs below, this is neutralizing titers done by the CDC for 3 different serotypes of IPV. The black is the bolus control, so 3 shots. And the blue there and the red, but I want to focus on the blue first, is our particles, so single injection. And you can see it didn't work very well. It didn't certainly work as well as the 3 shots, bolus shots. But then we reformulated and stabilized the IPV. And as you can see there in red, we have very exciting data showing that it's quite stable and creates the same neutralizing antibodies, these multiple shots. In fact, it even creates them sooner, which we think is related to some of the adjuvant activity of the material in the system. We've since then gone into non-human primates, which this data is in the lower, comparing it to our rat data. And we also have very strong responses, especially for type 2 and type 3 neutralizing antibody, which are the bottom right graphs. And again, this was all very encouraging for us, and this just summarizes the different type of products or vaccine products that we can put in these particles. Again, everything from recombinant proteins to inactivated vaccines. And of course, now we're doing a lot of work with mRNA, LNP-based vaccines as well, and have been successful in stabilizing them in these particles, and some data to illustrate that. And at least in the animal models so far, we can deliver an active dose of mRNA vaccines in our blocks. And then finally, I want to mention this technology was also used in several cancer applications. In particular, we use it to deliver STING agonist as an immunotherapy in cancer. And we've shown that, again, multiple injections versus our single particle formulation were studied, and our single particles not only did as good, if not better, but they've also shown that in certain cancer models, leaving the particles behind after tumor resection can decrease the number of metastatic cancers developing in the mice. And now we have a company who is developing this novel manufacturing, and hopefully will be in humans in the next couple of years. So we're very excited to see how this technology develops further. In our research setting, we're using this technology to really study how kinetics can affect the immune system. And in particular, we're working with collaborators to look at HIV vaccine development and escalating dosing, as well as malaria vaccines and some other novel vaccines that are studying some of the novel vaccines that are being developed. And in the last few seconds, I just want to highlight another technology, and then I will end with that. And this is based on microneedles. So we, in collaboration with BARDA, were able to develop a on-demand printer that could be shipped to different parts of the country or the world, if you will. And we actually started this work before this pandemic in an effort to try to have a tool if we did have a pandemic where we could ship these type of printers. And the idea is that the ink for the printers would contain the vaccine of interest. And the technology is based on microneedle technology. I don't know if anybody's familiar, but microneedles are these tiny little arrays that can fit kind of inside a Band-Aid that can be applied through the skin, and you could get a vaccine that way. This is one that could hold a human dose of a particular vaccine. So we developed these printers, which we pivoted during COVID and ended up using the mRNA LNPs encoding for the COVID vaccine proteins. And we were able to show that one can encapsulate mRNA LNPs in a very stable form in these microneedles, and they can be administered in the animal model and give immune responses equivalent to that of the regular bolus injections as well. This has no sharp waste, so once it's administered, it can be just like a Band-Aid thrown into the garbage. And again, the modularity of using a printer and shipping it to different locations, like a refugee camp or military base, can be quite advantageous. So with that, I'm going to end my presentation, and I'm happy to talk more if anybody has questions. But I just wanted to highlight some of the wonderful people at MIT that really make all this work possible, especially Tan and Nguyen, who developed the technology for single injection vaccines, and also Oriane van Straten, who helped develop the 3D printer, and of course my long-term mentor and collaborator, Dr. Robert Langer. So with that, I'd like to thank all of you for your attention, and again, I really appreciate the opportunity to present our work here today. Thank you.